Good afternoon, everybody. Can you hear me okay?
Yeah, I can hear you.
Great, thank you. All right, today we're going to start with chapter 25, then go to 23. Then we're going to do a quick review for the exam. We have no exam from last week to review, so that will save some time. So we'll do those 3 chapter sections and then our review for this week's test, and then we'll call it a day. Okay, we're going to get started here in just 1 minute. Okay, let's start with chapter 25A. Everybody see chapter 25A, renal? Okay, here we go. Function of the kidneys. What do our kidneys do? Well, we talked about how they filter blood, but the main thing that they do is they produce urine and remove the body of metabolic wastes. So they regulate extracellular fluid volume. They do this primarily by determining how much water is retained from the material that leaks from the blood into the glomerular tubules, and that is called filtrate. So the amount of water that's pulled back from filtrate is how you regulate extracellular fluid volume. Blood pressure in the same way. More water in the blood, more water in the body, higher blood pressure. Blood salt levels, it does this in a similar way. All of the salt is removed during filtration, and the amount that we want to keep is pulled back into the blood, and that's how we regulate how much salt we have in our blood. pH is done in a similar way as well. Certain ions are extracted or excreted, actively secreted, and some are retained. The main thing that we focus on with the kidneys is the removal of waste and foreign substances. The most common being nitrogenous waste, urea. You guys have heard of urea, and that's the main thing that is eliminated from the kidneys, in addition to other substances. Okay, the production of hormones. Erythropoietin is a hormone that controls red blood cell production, and renin helps control water retention, believe it or not. So it's actually going to be secreting a hormone that helps regulate itself, ironically. Okay, so a little bit of review of the anatomy of the kidney. Urine is produced in the nephrons of the kidneys. The process of that is called urination. What most people say urination is when they go to the bathroom and release urine. Physiologically, we're all urinating right now. You're probably not in the bathroom right now, but urination means the production of urine, not the release of urine. That is called micturition. So you can use different terminology now. Urination is production, and micturition is the release. Urine is conducted to the urinary bladder by the ureters. There's 2 of those, one in the kidney. Urine is stored in the bladder until it can be released, and that is micturition. Ourselves and other territorial animals tend to hold our urine until we find an appropriate spot. Animals like horses and most herbivores will just go kind of wherever they are. Micturition versus urination. I talked about micturition is the release of urine. Urination is the production of urine. So there you go. Urine is conducted to the outside of the body through the urethra. You have one of those, longer in males than in females. Okay, pretty straightforward. There's a quick picture showing you how it's set up. We have the kidneys, the suprarenal or adrenal glands. We have blood vessels, renal vein, and renal artery going in. And then the ureter is conducting urine from the kidneys down to the bladder. This is a female. And then the urethra goes from the bladder to the outside the body. Okay, this is an image from your text. It's a really nice detailed image showing you some of the details here. If this were anatomy, I'd be asking you to know the names of these parts. But what I do want you to know is that the functional units called the nephrons are found here between the renal cortex and renal medulla. So the cortex is the outermost part, the medulla is the center. These are called columns, and then pyramids, named so because they look kind of pyramidal in shape. The whole center part is called the cortex, the medulla. The outer part is the cortex. Okay, nephrons are the functional units of the urinary system. They filter the blood to form urine. Now remember, filtration is not about cleaning. It's just separating the liquid from the solids. The cleaning part of it comes from returning back the material you want to keep, the water you want to keep, the ions you want to keep, and the sugar you want to keep. Most of the nephrons are found in the cortex or outer portion of the kidneys. Most of the collecting ducts are found in the medulla. So most of what we're going to be talking about is in the cortex. And then the collecting ducts, which at this point is primarily fully formed urine, is going to be sending it down to the pelvis, which is the center of it, and then out through the ureters. Okay, this is a really good picture from your text showing you the anatomy of a nephron. So I will be asking you some questions about how this works, but a really quick rundown on how this works. If you see the glomerulus is the site where the blood is actually filtered. In this spot, filtrate, which is basically just plasma minus the large proteins, is going to leak out here and then make its way into these tubules. As it makes its way into the tubules, two main...
You paused. You paused for the last 10 seconds.
Uh-oh. What was the last thing you heard me say?
You said the glomerulus, and then it went out.
Oh, okay. Well, I didn't get an unstable internet connection image, so I don't know why. Okay. Anyway, so the glomerulus is the site where the filtrate leaks out of, is collected by the glomerular capsulae and makes its way into the proximal convoluted tubule. Proximal means near, proximal to the glomerulus. So here it's going to go through these tubes, and what's going to happen? You see these blood vessels around it. Two main things are going to happen. The body's going to reabsorb the materials that it wants, water, salt, sugars, and then it's going to actively secrete other materials into it. So the idea is, it's kind of, the analogy is kind of like, could you clean your house by taking every single thing out and then returning the things you want to keep? Well, it's not the most efficient way to do it, but that's how the kidneys work. It dumps all of the fluid into this tube and then takes back the stuff that it wants. Now, this is really amazing because what it means is the body doesn't have to know that something's a foreign substance. The idea is it gets rid of everything and takes back the good things that it wants, and then actively secretes the stuff that it knows it doesn't want, such as urea, back into these convoluted tubules. Now, in this dip here called the loop of the nephron, sometimes called the loop of Henle, some salt balance happens here, some more water is reabsorbed. And then in the distal convoluted tubule, the final determination of how the urine is going to be, how much water is in there, how much salt. And then by the time it gets in the collecting duct, it's urine and makes its way to the renal papilla, and from there through the ureters and to the bladder. Okay, so this is a figure that shows you, this was the anatomy, and this is the process of it. So remember, there are no more essays in the rest of the class and no more extra credit, but I may be asking you questions about this figure. So in this spot, filtration is where the liquid separates from the solids. About 20% of the liquid that goes through here leaks out. And then here you see R is reabsorption. This is going to be a reabsorption of water, ions, sugars. And then secretion is the movement of materials that the body knows that it does not want out of the blood and into the proximal tubule. You see more reabsorption happening here. This is important because the filtrate is becoming saltier, so it's going to take a salty environment to reabsorb water back into it. There's your distal tubule. I showed you the anatomy. The final reabsorption and secretion, it shows a little bit here, but there's virtually no reabsorption or secretion happening in the collecting duct. It's almost all completed by the time it gets to the end of the distal tubule. Now, this used to be an essay, but I took it off for the summer because you guys have been working really hard and you've been going really fast. So don't worry about this as an essay. All right, blood vessels, the nephrons, and the process of filtration. All right, afferent means towards. Now, when we were talking about the nervous system, afferent was sensory. That means towards the central nervous system. So here, Afferent means towards the glomerulus. And you can think of the glomerulus as kind of like the central nervous system. This is the spot of filtration. So afferent means towards, efferent means away. So filtrate is removed from the glomerulus into the Bowman's capsule. It leaks very much like those soaker hoses that you may have seen in gardening, where you put water in it, and water kind of oozes out of this of the soaker hose, and that's how it works in those capillaries of the Bowman's capsule. The efferent arterioles are then taking blood away from the glomerulus after a certain amount, usually about 20% has leaked out, and then it moves it into the paratubular capillaries, where you're going to take back a lot of that water and the good stuff that the body wants. Some water and salts are reabsorbed, while other wastes are actively secreted. I call it excreted because it's going into an excretion into the proximal tubule. So the main business of reabsorbing water and salts and moving waste out happens in the proximal tubule. All the rest of the nephron is just balancing, making sure you have just the right amount. For example, if you drink a whole bunch of water, you're going to have to micturate more or pee more. And that's because your body doesn't take back as much water when it produces filtrate. But if you're thirsty, you're still filtering your blood, but your body's going to take back most of the water. That's why when you're thirsty, your urine tends to be highly concentrated. And when you've been drinking a lot of water, it's more watery. And that's because your body's trying to get rid of the excess water that you've consumed. The majority of all water that is reabsorbed occurs in the proximal tubules. Okay, so that's a review question and test question. You might want to make a mark in your notes or a note in your notes that this is a test question.
For the sake of clarity,
Sure.
I was just wondering, is there any difference at all between micturition and urination?
Yeah, I explained that earlier. So we tend to say urination is the business of going to the bathroom, but physiologically, urination is happening continuously as long as you don't have renal failure. So all of us are urinating right now. That means the production of urine, and that's then going into our bladder. Micturition is what happens when your bladder becomes full and you go to the bathroom and you excrete it, you release it from your bladder. So urination, constant production, micturition, the release in the bathroom, or onto the ground if you're doing camping style like my brother likes to do.
Thank you.
All right, does that make sense?
Oh, yeah, perfectly. Thank you.
Okay, loop of Henle. This has a slightly different name in the image that I showed you. It was the nephron loop, but when you see that loop, it means that dropping down, the thinning, dropping down. What's happening here is water and salt are reabsorbed into the loop. So because the filtrate is becoming saltier, you need a saltier environment. And so the loop of Henle has very salty cells. What that does is through osmosis, the water goes into that salty environment, pulling more water out of the filtrate and helping you retain water. By the time the filtrate leaves the loop of Henle, LOH loop of Henle, it's lost 90% of its original volume. So by the time it leaves the loop of Henle, there's only 10% of what leaked out originally left. At the distal tubule, salt and water balance take place as a result of hormones. So hormones determine, do we want to retain more salt? Do we want to retain more water? Or do we want to let it go because we're consuming lots of fresh water? When filtrate enters the collecting ducts, it is urine, and generally nothing else happens to it. A tiny amount of reabsorption and secretion can occur, but by the time it gets into the collecting ducts, it's about 99% finished. And then when it gets in the bladder, it cannot be further modified. In addition, it cannot be further modified either in the renal pelvis or in the ureter. So once it leaves the collecting ducts, at the end of the collecting ducts, nothing more changes about the urine. Okay, have a funny animation here. I don't know why that happened, but off we go. Okay, the process of, whoops, I must have gone the wrong way. Okay, filtration. We leak about 180 liters per day are filtered, but only 1.5 liters on average are lost in urine. So what does that mean? The vast, vast majority of what leaks out of your capillaries into the glomerular capsule gets pulled back. Now, if it didn't, you would die pretty quickly because that's way more than your entire bodily fluid. So what it's telling you is that your body fluid is being, all of your fluid and blood is being completely filtered many, many times per day, in the same way that a aquarium filter filters many hundreds of gallons of water, even though your tank is only 20, 40 gallons total. Filtrate is similar to plasma, but is missing the large plasma proteins, which are globular proteins called globulins. The filtrate is composed of water and dissolved solids. Cellular components are not filtered. So when we say cellular components, we're talking about things like red blood cells, white blood cells, thrombocytes, which are platelets, and the large plasma proteins are not released into the filtrate. So they are retained. So when it says they're not filtered, it means they are held in the blood, not allowed to become part of the filtrate. Okay, this is an image from another book that I really like. This shows the glomerulus, and what we see on top of it are cells called podocytes, and that means foot cell. And it looks kind of like alien feet that are interlocking with each other, kind of like fingers interlocking. What that does is, since these holes in the capillaries are not variable in size and fluid can leak right through, these filtration slits, which are the gaps between these cells, can vary, and that can determine how much fluid leaks from the blood down into the foot, past the foot processes and into the Bowman's capsule to become filtrate. Okay, this is a picture showing you the size of these holes. Now, I don't know if you guys have ever looked through a microscope under a red blood cell, but they're really, really small. So if this picture shows an erythrocyte or a red blood cell looking really big at the bottom, what that shows you is that these filtration slits are very tiny. And they have to be so tiny that the plasma proteins in the plasma do not make it into them. Now, if they do, then you have proteinuria, and it's an indication of a problem with your kidneys. So proteinuria means that proteins from the plasma are making it into your urine, and that can be tested for in a urinalysis. Filtered plasma must pass through endothelial pores and filtration slits. The filtration slits are the slits between the podocytes. The foot processes of the podocytes wrap around the capillaries of the glomerulus, forming slits like I showed you. And this is the direction of the movement of the filtrate. This is one of my favorite things to say in the class. Slits form small gaps through which filtrate flows. Try to say that 10 times fast, you probably get stuck. But there it is. That's how the filtrate is formed. All right, this is another picture showing a scanning electron micrograph, showing you the podocytes, the pedocells, which are the little fingers of these podocytes. And you can see the little gaps right in between these podocytes are the filtration slits. Okay, hydrostatic pressure, what we call blood pressure, causes filtration. So the production of filtrate happens as a result of the fact that your blood is under pressure. You guys know that we have a certain amount of blood pressure, and the pressure in the nephrons is functionally zero. Now, this is interesting because our bladder can get full and under pressure, but we always want to maintain a very low pressure in the nephrons. And I'll show you how that works coming up. The hydrostatic pressure of blood, or blood pressure, causes the fluid to leak through the gaps in the glomerular capillaries in the same way that the leaker hose leaks because there's pressure in the hose and less pressure outside. Some fluid moves back due to colloidal osmotic pressure. I talked about that when we talked about bulk flow and reabsorption in the capillaries. The presence of the plasma protein adds osmotic pressure, which pulls some of the fluid right back. Only 20% of the blood plasma is lost to filtrate. The process produces 180 liters per day. So what that means is we're filtering our blood many, many times per day so that our blood is constantly clean. Entire blood plasma is filtered 60 times per day, or 2.5 times an hour. This is significant, especially for people who are going to go into renal treatment centers. What is the name of my brain just blanked for a second.
Dialysis?
Dialysis, thank you. Dialysis centers. So dialysis centers are interesting because the patients usually wait 2 to 3 days on average to get into a dialysis clinic. And what that means is they're accumulating toxins and fluids, et cetera, for 3 days. And that what that means is if we're doing it 60 times a day and they're doing it 1/3 of a time per day, that means that the amount of material building up in their blood is pretty significant. And so when they go in, what's going to happen is a lot of fluid is lost, a lot of wastes are lost, a lot of salt is lost potentially. And what can happen is people can code. And coding means that your heart stops. Now, one of the discussion questions I guess you guys didn't get was, what would you do if somebody coded and had a tattoo on their chest that said "Do not resuscitate," but didn't have a DNR on file. Would you resuscitate them or not? And
Well,
my
that was actually one of our discussion boards.
It was a discussion. Okay, so you guys have had a chance to discuss that. Many of you said, well, you would resuscitate them anyway because a tattoo on a chest saying "Do not resuscitate" is not the same thing as a DNR on file. So it's kind of interesting. Would you want to take or what if someone had told you verbally, "I do not want to be resuscitated," but they did not have something on file? Would you do it or not? It's an interesting ethical question. And I knew someone who did work in a dialysis clinic, and several of her patients did code or die, and that was it. They had DNRs and they didn't want to continue. So it's an interesting ethical question. Okay, so this is showing you the process of how material leaks out. So notice the proteins here are green, they're retained, and the fluids leak out because there are gaps there. Now, the interesting thing about reabsorption is there is some energy required for reabsorption, but there's a little trick that the body plays, and let's take a look at how it works. So all of this material, salt and water, leaks out. Now, how do we get the salt and water back? Well, all we have to pull back using active transport, that means ATP energy, is the sodium. Because the chlorine is associated with the sodium, it comes passively along with the sodium. So that doesn't take any energy. Now you have salt on this side and water on the other side, so water follows salt by osmosis. So in order to get both water and salt back from the filtrate into the blood, all you have to actively move is the sodium ions, and the other two materials come back passively. So that's kind of interesting. Now notice fluid is reduced to 1/3 of the original volume, but it's still isoosmotic. What that means is the amount of salt on both sides is equal because you're moving sodium, chlorine, and water all out. So all it's doing is reducing volume, not changing the osmolarity or osmotic force of the fluid in there. Okay, the effect of blood pressure on filtration. Now, blood pressure provides the hydrostatic pressure that drives filtration. We need it. What's interesting is you might guess that as our blood pressure goes up, we would filter our blood more. As our blood pressure goes down, we filter it less. It turns out that's not really the case, and I'll explain why. Changes in blood pressure do not directly affect filtration rate. Now, this seems counterintuitive because doesn't blood pressure cause filtration? What happens is in the kidneys, it kind of maintains a little local environment in which the pressure of the glomeruli, glomerulus, remains constant even under fluctuating pressures. So glomerular filtration rate, that means how much liquid is flowing from the blood into the capsule and then into the proximal convoluted tubule, is constant under a variety of pressures. So when your blood pressure goes up or when it goes down, the GFR remains constant because your body maintains a local pressure that is constant even though your overall pressure fluctuates. And I'll show you how the body does that here. So vasoconstriction and dilation in the afferent and efferent arterioles balance changing blood pressure. Now, let's talk about something kind of unrelated, which is male erections. So how does an erection work? Well, when a male becomes sexually excited, what happens is the arteries that go into the penis open up. That allows blood in. Now, to make sure that the pressure increases locally, what happens is you decrease the outgoing pressure or venous side, and that causes what is called vasocongestion. And that maintains more pressure in the penis than there would be in the regular body. And so the erection happens. So in a similar way, the body can increase or decrease the local pressure in the glomerulus by opening and closing incoming and outgoing. So if you close off incoming and open up outgoing, the pressure would go down. And if you did the reverse, which is open up incoming and reduce outgoing, in the same way an erection happens, you would increase the pressure in the glomerulus and the glomerular filtration rate would go up. So let me show you how that works. So this arrow shows you GFR, or glomerular filtration rate, and it shows both of them open. So if you increase the resistance coming in and open, or basically just increase the resistance on the incoming, this would be the afferent, you notice that GFR decreases because the hydrostatic pressure, so HP, pH as hydrostatic pressure drops, and that means GFR drops. Given a normal blood pressure. In this case, if you do the opposite, this is what's called vasocongestion. You decrease outgoing, maintaining ingoing, the hydrostatic pressure here rises, and you increase GFR. Now, what's more interesting is what we're trying to do in the body is not to increase or decrease GFR, but maintain it. So what would happen is you would do C if you wanted to increase or maintain GFR under low blood pressures, that is when you're sleeping. And you'd want to do this under really high blood pressure, let's say during exercise. So if your overall blood pressure shot up, you would do this, and that would maintain GFR nice and steady. If your blood pressure dropped, like during sleeping or rest, you would do this, which would maintain GFR. So this figure is showing what would happen under normal blood pressure if you did these things. But what's more interesting is how does the body maintain this GFR steady under fluctuating pressures? So there are definitely going to be questions about these figures and ask, what is going to happen to GFR when this happens? What is going to happen when this happens? And this one is kind of a mystery. You don't need to worry about it. In this case, GFR drops. In this case, it increases, and this is where it's nice and steady. Okay, so there may be pictures about it on the test for that. Okay, so let's go to 25B, which is more renal. Okay, hormonal control of renal function. Okay, autoregulation of GFR. So what does autoregulation mean? Now, do you guys remember early on I talked about a really important concept? It was called homeostasis. And homeostasis means making sure everything stays nice and even. Now, one of the ways the body does, and the main way the body does homeostasis is through a process called negative feedback. That means if you get out of normal, the body responds, causing some effect. And then when you get back to normal, the feedback back to what is causing the change becomes negative. That is, we're no longer out of normal. And that says stop doing the change that was trying to get us back to normal. That's called negative feedback. And what's interesting about the kidneys and the nephrons especially is that negative feedback becomes right next door. So it doesn't have to go a very far distance because look, here's the distal tubule of one, and here's the proximal, the afferent and efferent arterioles for that very same nephron. So what it means is if there's a large amount of fluid flowing through this distal tubule, that can affect afferent and efferent arterioles. So what happens is negative feedback is immediate, right next door. So if the flow is too high, you would decrease afferent, increase efferent, and if the flow is too low, you do the reverse, and that's right next door, right immediately next to it. So these modulations of these flows can be instantly and quickly and automatically controlled. Okay, so this is called the myogenic response. It's the intrinsic ability of vascular smooth muscle to respond to pressure changes. Vasoconstriction in the afferent arterioles occurs as a result of increased blood pressure. So this is what we were talking about. So we increase constriction, that means decreasing the flow into the glomerulus if blood pressure rises. When this occurs, blood flow is reduced, causing maintenance in GFR. So it's the opposite of what we saw in the figure. Severe decreases in blood pressure cannot be compensated for, and GFR will decrease. So for example, you go into shock. If you go into shock, this process of opening up incoming and decreasing outgoing doesn't work anymore. And as a matter of fact, you don't necessarily want to be filtering the blood if you're functionally dying because your blood pressure is dropping. You guys have probably seen the shows that have medical drama in them, and they say, doctor, the patient's blood pressure is dropping, and that usually means that that's a problem that has to be addressed by the physician, or you potentially would lose the patient. So if you drop too low, that business of increasing the local pressure stops, and you stop filtering your blood until you get back to some normal blood pressure. So this figure is showing what is called tuboglomerular feedback. So tubo, the tube, glomerulus is a mechanism through which changes in the fluid flow through the distal tubule influence GFR. So as I said, if the flow through here is too high, you decrease afferent and increase efferent, which means that the leakage is going to decrease. And if the fluid flow is too low, You're going to do the opposite. You're going to open the afferent, close off the efferent, pressure in here is going to go up, and then eventually the fluid volume and flow will increase. So this is the way the body does it. What it does is it helps maintain the homeostasis of glomerular filtration rate. Okay, hormones and the autonomic nervous system on GFR. Angiotensin is a hormone that causes vasoconstriction. Prostaglandins cause vasodilation. So these are the two hormones that primarily are going to affect the vasodilation, vasoconstriction of the afferent and efferent arterioles, helping maintain GFR. Both afferent and efferent arterioles are innervated by the sympathetic nervous system, which is normally responsible for fight or flight. If blood pressure drops severely, blood loss or dehydration, the sympathetic nervous system causes vasoconstriction of all renal blood vessels. So now why would it do this? What it's saying is not only do we not need to filter our blood now, but our blood pressure is dropping. And remember that as we filter blood, some fluid moves from the blood into the tubule. And if your blood pressure is dropping, you need to maintain all of the fluid that you can, even if it causes you to retain some toxins or waste. The idea is you don't want to filter the blood if you have very, very low blood pressure, because you need to maintain all the fluid on the arterial side in the blood as possible. Then if you get back to some normal, the sympathetic nervous system will shut off, and then the kidneys go back to filtering the blood again. So this causes a decrease in GFR. So somebody who has really, really low blood pressure is not going to be producing a large amount of urine or filtrate. Okay, reabsorption. About 99% of all filtrate must be reabsorbed. Most of the reabsorption takes place in the proximal tubule. I talked about that earlier in the lecture. Only water and ions that are needed are reabsorbed. The rest are lost to urine. So that's the idea. You dump everything, take back only the stuff you want to retain. Active reabsorption, most water and salts are taken back into the blood. ATP is needed. Remember I showed you the only ATP that's needed is for the sodium. Chlorine follows passively, and then through osmosis, the water molecules follow those two ions. Okay, active secretion, which is a form of excretion because you're moving it into the urine. Secretion or excretion into the lumen is the transfer of extracellular fluid into the lumen of the nephron. So what's happening is the body is identifying materials that it perceived to be a waste and moving it from the blood actively into the filtrate so that you can get rid of it faster than would be the case by just letting it leak out due to the production of filtrate. This is important for materials to be eliminated quickly by the body. So penicillin, when it was first invented, I'm sorry, not invented, it wasn't invented, it was discovered as a way to battle bacterial diseases. What they found though, the problem was that patients were excreting it really quickly and it wasn't allowed to maintain in the blood very long, and so it wasn't working on killing the bacteria. So when first discovered, all penicillin was filtered out of the blood within 3 to 4 hours of administering. You don't want it to be lost quickly because it can't do the job of killing the bacteria. So this chemical was added to it called probenecid. It reduces the active secretion or excretion of penicillin by the kidneys, allowing it to last longer in the blood. And this is done now for most antibiotics. There's a substance that blocks, it actively competes with the antibiotic, because even though the body identifies the antibiotic as a foreign substance that it believes it needs to get rid of, by having this kind of fake material take the place of the antibiotic, it allows the antibiotic to retain a half-life or a total, I can't remember the name it is when your body has removed half of it from the blood. It's like a something 50. You guys know what I'm talking about? Anybody know that one?
No, I think that's called the half-life. The half-life.
Okay, I was thinking about the half-life of radioactive elements, but I guess we'll go with half-life, which is how long it remains in your blood. But it's usually measured when about 50% of it is gone. So that, I guess the half-life makes sense. All right, micturition. Smooth muscles move the urine from the renal pelvis through the ureters into the urinary bladder. Now, remember I told you I was going to tell you how do you maintain high pressure in the bladder and really low pressure in the rest of the urinary system. It does this by pushing the urine actively down through the ureters, pushing it into the bladder, and then closing quickly the sphincter muscles so that you're kind of like squeezing material along a tube, opening up a little valve pushing the fluid in and closing the valve again. So what that does is it maintains a low pressure in the nephrons or the renal system and potentially having high pressure in the bladder. Now you have to maintain really low pressure in the nephron, because what's causing fluid to go from the blood into the nephron, the glomerulus, is the difference in pressure between blood pressure and the pressure in those tubules. If the pressure in the tubules rose, then GFR would stop and filtration would stop and your kidneys would stop working. So that's why your bladder keeps getting more and more and more full, and your body assumes you're going to be able to micturate pretty soon. And then if you don't, either your bladder can rupture or you'll pee yourself because you can't hold it in. All right, urinary bladder is surrounded by smooth muscle. That is unconscious. So when we micturate, what we're not doing, we're not contracting the smooth muscle around the urinary bladder. We're relaxing skeletal muscle, that's the sphincters that surround the urethra. And then when that happens, a reflex occurs, and then the smooth muscle surrounding urinary bladder starts to squeeze. Micturition is the voiding or release of urine to the environment. There are two sphincter muscles controlling urine release: the external, which is conscious, that's skeletal muscle, and the internal, which is smooth muscle. So basically you relax external, then what follows that is the internal, and then the smooth muscle around the bladder. Some people are in public restrooms, especially some guys, have what is called stage fright, and that means that you relax the external, but the other don't relax because you're feeling threatened. And it is kind of strange to go to micturate around strangers, but that can happen in public toilets. All right, micturition reflex cannot be inhibited in pre-toilet trained infants. So the micturition reflex means you have to wear a diaper. So who else wears diapers besides infants?
The elderly.
The elderly. Now, when the elderly are wearing a diaper, does that mean they have a pathology that needs to be seen by a doctor? Or is that part of the normal aging process for some people?
Sometimes it does need to be evaluated by a provider.
Potentially, but for most people who end up in depends or undergarments, there's not much that the medical provider can do, right?
Well, yeah, like, because I remember after I had my baby, and I've heard this with a lot of other women too that have had kids, that the baby coming through the birth canal ruins the sphincter and the bladder. So if you cough or laugh, you'll urinate a bit. Yeah. Right.
So, and that happens as a result of some nerve damage happening as a result of the stretching of the vaginal walls, potentially. Okay, so you just answered a really good question. I asked on the test, what 3 people would you expect micturition reflex not to be able to be inhibited? And so it says in the notes, pre-toilet trained infants, but we just brought up 2 more people: the elderly, and that sometimes nothing can be done about it, and some women post-pregnancy. Some post-pregnancy women have have to wear some pads or undergarments, and that does not mean it's a pathology that necessarily you need to see a doctor for, but either something that you get better over time, or it's just a change that happens, and that then you would have to wear, like what my ex used to call pee-pee pads, and she for the rest of the time had to do that. And so please note, those are the 3: pre-toilet trained infants, some elderly people, and some postpartum moms. Okay, so there you go. You heard it from me. Most adults can suppress the reflux until appropriate time and place can be found. Okay, so most of us do find appropriate places to micturate. All right, and that's it for that chapter. Okay, so let's get to our last chapter, which is chapter 23.
Cool. Digestion.
Okay, digestion is
Okay.
next. I'm glad I haven't had too many unstable internet connections. I'm actually using my phone as a Wi-Fi source because my home internet really sucks. But it seems to be working, so I'm happy. All right.
Yeah, your hotspot seems a lot more reliable.
Yeah, but I'm not pushing it by having my own video on. That's why you see my static face. But I guess seeing my face doesn't really help the lecture. You know what I look like. All right, chapter 23, digestion. Here we go. This is a figure directly from your text. This shows you the anatomy of the stomach. It shows you the different parts, the fundus, the area that comes up above. We also find that in the gallbladder as well as the uterus. And so what is the fundus? It's basically an area that rises up above the entrance of the structure, the tube being below and then the rising part above that. So what we'll see in reproduction is the fundus is the area below the oviducts where the oviducts enter. And the same thing with the gallbladder. So you're going to see that same structure. The fundus is a rising up of a bag-like structure below the entrance of a tube. So that's why it's called the fundus. The body is the whole central part. The pylorus is this narrowing. And then this is called the pyloric sphincter. It's the valve that allows some food mixed with digestive juices to enter the duodenum. At that point, it's called chyme, C-H-Y-M-E. It's no longer called food. So once it leaves the stomach and enters the duodenum, which is the first part,
I think you spoke too soon about the internet connection. You just cut out.
you go. My internet.
You cut out pretty bad just now.
Are you still there, Professor? Oh, we completely lost you.
Oh.
Can anyone hear him?
No,
No? Okay.
it looks like he completely dropped off. He's not even on the roster now.
Yeah, I don't even see the PowerPoint anymore either on my screen. I wonder if we should exit out and reload the meeting.
I don't know.
I don't know if there's a way for him to come back in, is there?
Well, he was the one that made the meeting, so he should have a code.
Yeah. I'm going to try to exit out and just go back through the modules and come back in and see if maybe that reconnects anything on my end. But maybe I'll just end up right back here with you.
It looks like he's coming back on.
Can you hear me?
Okay.
Can you
What
hear
was
me?
that?
Yeah.
Yes, we can hear you now.
Oh, I just got done talking about how great my hotspot was and it just quit on me.
Yeah, we know. I
You
think
jinxed
So
yourself.
now I'm back onto home internet. I'm onto Wi-Fi. So we'll see how the rest of this goes. I'm glad it didn't stop the recording. So there's just going to be a weird moment in there where people watching are going to be like, well, does he ever come back? And I do. All right, let's do this again.
Take two.
All right. Slideshow from current slide. All right. What did you guys hear me say when it came to the stomach?
You were talking about the sphincter and then you cut out.
Okay. I'm going to do this one last time because I'm nothing if not persistent. All right. So when you mix food digestive juices, some of it goes into the duodenum, which is the first part of the small intestine. When it leaves the stomach, even in the stomach, when it's mixed with digestive juices, it's no longer called food. It's called chyme, which is C-H-Y-M-E. So everywhere from the duodenum down to the large intestine, it's called chyme. When that material enters the large intestine, it's now called waste or feces. So there you go. Okay, so this is another picture from your book that shows you what the inside of the stomach looks like. We can see gastric glands. We can see parietal cells, which are going to be producing some of the substances, and enteroendocrine cells. We're going to be going into what they do. But basically, these layers of the mucosa are going to be producing both a bicarbonate buffer to protect the surface of the stomach, as well as the hydrochloric acid and the mucus that is, well, I kind of mixed those up. The hydrochloric acid is going to be doing something other than what you would expect. And you would think that the acid would be digesting the food. It doesn't. It actually turns out the acid that's produced by these chief cells and parietal cells is going to be denaturing proteins. Now, what is the significance of denaturing proteins? It does two things. Hydrochloric acid both kills bacteria and viruses and any other invading organism by denaturing their proteins. But what it also does is any proteins in your food, because they have a three-dimensional structure, it causes them to unfold and open up. And by unfolding and opening it up, it allows the proteases, which are enzymes that digest proteins, to get in there and break it apart to help you digest it. So here's the deal, guys. Hydrochloric acid, the acid in your stomach, doesn't directly the food. So when it gets into your small intestine, it doesn't
Pastor,
cause.
can you repeat that again? You started talking about the hydrochloric acid and then you cut out.
Goodness, I'm just not having a good internet day. Okay, one more time. The hydrochloric acid does two main things. It kills bacteria and viruses and denatures proteins. That causes them to unfold, allowing the proteases, which are enzymes that digest protein, to get in there and break them into amino acids. So even though hydrochloric acid doesn't do any digesting, it does do an important job. In addition, it allows certain enzymes in the stomach to be activated. For example, pepsin is activated by the acid. So if I ask the question, does hydrochloric acid digest anything? The answer is no, it does not. It just kills invading microorganisms and causes denaturing of the proteins to assist in the digesting process. Did you guys get all that?
Yes.
Awesome. Okay, here we go. So oral cavity is your mouth. It's the site of mastication, which is a scientific term for chewing and swallowing. Salivary glands produce saliva containing salivary amylase. Salivary amylase is an enzyme that helps you begin the digestive process of carbohydrates. So the question is, why do you need to digest carbohydrates in your mouth? Two really important reasons. First is it allows you to identify carbohydrates as being made out of sugars. The idea is if we break carbohydrates down, it produces simple sugars, and the simple sugars taste sweet, and that allows you to know that the starch is containing sugars that's good for you to eat. In addition, it helps clean your teeth because starches tend to stick to your teeth, and by producing salivary amylase, it dissolves that starch that's stuck to your teeth, washing your teeth, reducing bacterial dental caries, which is cavities. Okay, so the salivary amylase helps clean your teeth as well as begin the digestive process so that you know the starches contain sugars, which is energy. All right, the next stage is the esophagus. It connects the mouth to the stomach. Really, the whole point is it takes food from your mouth through your thoracic cavity, past your heart and lungs, to your stomach, which is below that. The stomach, the fundus, I talked about the upper portion is attached to the esophagus. The body is the main or middle portion. The rugae are the inner folds. It works kind of like pleats on a skirt or on a curtain. It allows stretching out so that the mucus and bicarbonate buffer underneath the mucus can be, the integrity can be maintained so the acid doesn't start digesting the stomach lining. The antrum is the lower portion attached to the duodenum, and the end of the antrum is called the pylorus that contains the pyloric sphincter. In most books, the antrum and pylorus are put together, being called the pylorus. The antrum is not normally separated out. Okay, more anatomy. Small intestine. The first part of the small intestine is called the duodenum or duodenum. Those are both correct pronunciations. The first part, it receives chyme from the stomach and digestive juices from the pancreas. In addition, it receives bile from the gallbladder to help you with fat digestion. The jejunum is the main portion of the small intestine. Most digestion and absorption occurs there. So please note, the main job of the stomach is not to digest food. It's to store it and to sterilize it. It's to make sure that all the food from your plate ends up inside you so you can take your time digesting it. It's not to digest food. So the digestion and absorption of food happens in the small intestine. The jejunum is the first and largest part of the small intestine. The ileum is the second or final portion. It's attached to the cecum, which is the beginning of the large intestine. Oh, I asked the question, what would you call the valve that joins the ileum to the cecum? It's called the ileocecal valve. Or I think the question says, what does the ileocecal valve join? What it joins is the ileum to the cecum. That's why it's called that, the small, the large intestine. The pancreas produces digestive enzymes in addition to hormones. The liver produces bile for lipid digestion. So the liver produces the bile that goes into the gallbladder. When you eat, Lipids or fats, it goes from the gallbladder into your duodenum. And what the bile does, it helps break the fats from large chunks into smaller ones so that you can digest it. The large intestine, the colon, is primarily for the removal of water from the feces and vitamin synthesis. It turns out that if you have an unhealthy colon, you can have a variety of health problems. So having normal, healthy flora in your large intestine is important. And it turns out if you don't have a healthy one, you can get what is called a fecal transplant. Sounds pretty nasty, but the idea is you're trying to get back to healthy bacteria into your large intestine because those are going to be producing vitamins, keeping yeast out. You don't want to have a yeast infection in your colon. And so having a healthy microflora in your colon is important to health. There's a lot of articles talking about that. If you're interested, check it out. The rectum is the end of the large intestine. It's for the compaction and storage of feces. And then the anus is the exit to the digestive tract. Okay, this shows you a picture of this directly from your book. The duodenum is the beginning of the small intestine. This is where the stomach would be. The jejunum is the first or main part. The ileum in red is the secondary part. And then the small intestine, they join up right here where my pointer is. You can't see the junction part, but the food or the chyme goes from the ileum, which is small intestine, into the large intestine. At that point, it goes from being chyme into waste. And then when it gets down to the end of the descending colon, it's now called feces. All right, so these are two pictures showing you how motility works, how we move stuff through our digestive tract. So peristalsis, this is the top picture. This shows you pushing food downward. Now, this is why it takes longer for food to get from your mouth to your stomach than liquids, because you actually have to squeeze a ring of contraction to push the food down. It takes a moment. Liquids shoot right down because there's very little problem. So this is how we push a bit of food or of chyme through the digestive tract by contracting the muscle on the proximal side and relaxing it on the distal side. That pushes the food down. But what if we don't want to move the food? What if we just want to mix it? So for example, we have these different clumps of food or chyme. If we want to mix them to make sure that the digestive enzymes get fully mixed and that the food gets fully digested and absorbed, this is what you do. You pinch it off. It's kind of like, I don't know if you guys have ever tried to mix something inside of a bag. You just kind of squeeze it various places and that causes mixing to occur. So instead of pushing it along, you're just pinching right in the middle. So opening between them, pinching in the middle, that causes them to mix and eventually go from blue and yellow to green. And that shows that good mixing has occurred. This is important because the only place that you can absorb food molecules is right on the surface of the small intestine. So whatever food molecule is in the center of the lumen of the small intestine doesn't do you any good. So you have to move it, keep moving. So it can be finished being digested and absorbed right there at the surface. What was the last thing you heard me say?
You have to keep moving it.
You have to keep moving it so that you can fully digest and absorb it, okay? All right, chemistry of digestion, carbohydrates. Simple carbohydrate foods are called sugars. Complex carbohydrate foods are fiber and starch. Most people know that complex carbohydrates are better than simple carbohydrates or simple sugars. And the question is why? Why is it better? Well, it's better primarily because it takes time to digest carbohydrates. And as you digest the carbohydrates, simple sugars, monosaccharides or disaccharides become released and they enter your blood. So what's the difference? Well, when you eat simple sugars, there's no digestion required and it quickly surges into your bloodstream, stimulating your pancreas and potentially leading to diabetes. But when you eat complex carbohydrates, the digestion takes longer, the sugar moves more slowly, doesn't cause the pancreas to be stimulated as much, and there's less stress on it, decreasing the probability of type 2 diabetes. Okay, enzyme hydrolysis. Hydrolysis means breaking down water to break down these molecules, so break down using water. Hydrolysis literally means hydro, meaning water, lysis means to split, so you're splitting water to split these food particles apart. Amylase, as I mentioned before, digests starch into smaller disaccharide subunits. Now, these disaccharides can be absorbed or further digested and then absorbed. Maltase is an enzyme that you find there. It digests maltose into glucose. Maltose is a disaccharide with 2 glucoses in it. So maltase breaks maltose into 2 glucose molecules, which can then be absorbed into the blood. Sucrase is an enzyme that breaks down sucrose. This is table sugar. When you add sugar to coffee or to tea, that's sucrose, and sucrase breaks it down into both glucose and fructose. So sucrose, the sugar, is made of 1 molecule of glucose and 1 of fructose. Lactase digests lactose into glucose and galactose. Some people don't produce it as much, and it's not surprising because if you think about it, the only people that are supposed to be consuming milk are babies. But our ancestors decided that cows and animals like cows would be a good source of food. That is, if we took their milk and made it into cheese or drank it, well, that would be a great way to supplement our diet. Well, it turns out everybody before the domestication of cattle and other ruminants, they're called, didn't have to consume any milk products after they were babies. And so really we had no need for lactase as adults. So when people have lactose intolerance, it's a completely normal thing because for most of our evolutionary history, humans did not consume milk after we were toddlers. So that's why lactose intolerance is understandable because most adults are not supposed to be consuming dairy products in terms of our biology. All right, so here's the starch glycogen. What we see is when amylase gets a hold of it, we end up with several different types of disaccharides. There's maltose, turns into glucose. There's sucrose, turns into glucose and fructose, and lactose turns into glucose and galactose. So this is carbohydrate digestion. Okay, we don't want to do that. Are we going to be able to get that slide on there? There we go. I don't know why it auto-animates, but anyway, proteins are composed of chains of amino acids. Individual amino acids here are shown as blue circles with the amino terminal end and carboxyl terminal end. If you've ever drawn a molecule of an amino acid, they have this part on one end and this part in the other with an R in the center, which means that it is, well, carbon in the center and then an R, which is variable. Anyway, so the amino terminal end is going to be attacked by one kind of enzyme, and then the carboxyl terminal end, named because the carboxylic acid is what it's primarily made out of. Okay, so let's talk about the different enzymes. So endopeptidases break it in the middle of the chain, forming 2 larger or smaller chains. But aminopeptidases are named because through hydrolysis, they break off what is called the amino acid on the amino terminal end. That's what I'm trying to say. And carboxypeptidases cleave it off on the carboxyl terminal end. So these are different than these, but either way we get individual amino acids. But it is important to note that even large proteins can be absorbed, and this is what leads to food allergies. And I'll talk about how food allergies work here in just a moment. Okay, we're going to do this one more time. Okay, fats are composed of both fatty acids and glycerol. Glycerol is the backbone. 3 fatty acids are normally held together by the glycerol. Most fats are hydrophobic, means that they don't mix with water. So what bile does, coming from the liver and stored in the gallbladder, what it does emulsifies or breaks the fat into small water-soluble fat droplets. Now, the reason why this is important is because our ancestors didn't have too much food like we do. They had too little. And if you couldn't digest the fats that were in your food, you could potentially starve to death. So bile is important biologically to make sure that we digest all the fats that are in. Even we need some fats because there are some fat-soluble vitamins such as A, K, and D that we wouldn't get otherwise if we didn't break down some fats. Okay, so the enzymes that break down fats are called lipases, colipases, and phospholipases. Phospholipases break down phospholipids. Triglycerides are broken down into monoglycerides and free fatty acids. A triglyceride means that there's 3 fatty acids attached to 1 molecule of glycerin. And you can see it here. This is called the triglyceride. The triglyceride is made up of the glycerol, which is the yellow, and 3 fatty acids. It shows them as a zigzag because as the carbons attach to each other, it kind of changes direction. If you guys have ever had any biochem or organic chemistry, pretty much biochem, you understand why it does this. But when the lipases and colipases break it down into a monoglyceride, which is a fatty acid and glycerol, and free fatty acids. Okay, I really like this figure. It is not in your book, but it does show you what's happening at each of the parts of your digestive tract, what's happening in terms of motility, the movement, secretion, what materials are secreted in, what digestion occurs there, and then what absorption occurs. So this is a great overview of what's happening in different parts of the digestive tract. This is not in your OER text. This is in your OER text, and it does show you a little bit of what's happening, not quite as good as the previous figure. You can see a lot more detail here, but this is what is in your OER text, showing you what is happening in different parts of the body. So propulsion, where that's happening, where chemical digestion is occurring, where mechanical digestion is occurring, and absorption. So instead of talking about each area, it's basically giving you a key to what the different colors of the arrows or the drops. Okay, cephalic and oral phases of digestion. Cephalic is the anticipation of food. What that means is when you start thinking of food or someone talks about food, you start producing saliva. That means you're preparing to digest it, and that is part of the digestive process. So when you get hungry and you start thinking about food, your stomach starts to rumble. Now, why does that happen? You start producing digestive enzymes. There's a little bit of gas in there, and you start squeezing your stomach in anticipation of eating. That's where the rumbling stomach happens. GI secretions begin before food arrives, anticipating the food getting there, so that when you do eat, there's already digestive enzymes in your stomach. Saliva is produced in anticipation of food. And then if you smell food, of course, you're going to salivate a lot, even especially sour food. Sour foods like what am I thinking of? Vinegar. You smell a pickle, you're going to produce a lot of saliva. So what happens in your mouth? Chewing, a scientific name is mastication, that starts digesting. What is the purpose of chewing? Is to break the food into smaller bits. That increases the surface area and allows the enzymes to better digest the food if it's in smaller bits than in larger bits. So grinding the food breaks up food pieces to increase the surface area for digestion, as I just said. You also are trying to mix the food with saliva, allowing some of those enzymes to get in there. But more importantly, you want the food to be slippery so they can get all the way down your esophagus and not get stuck. If it does get stuck, most people drink a little water to help it on its way. Saliva contains what? Water for liquefaction, enzymes to help begin digestion, mucus, which helps it become like a slip inside, getting the food all the way down to your stomach, and immunoglobulins, believe it or not, that help kill bacteria. The swallowing reflex. Swallowing itself is called deglutition. You guys see the word glut from gluttony? It means to swallow. So gluttony means you swallow too much. Pressure against the uvula begins the reflex. That's that little dangly thing in the back of your throat. The glottis rises and epiglottis covers the windpipe. The glottis is the top of the windpipe itself, and the epiglottis is the little leaf-shaped cover that covers the windpipe. If you want to see how that works, watch yourself swallow in the mirror. You'll see your Adam's apple, which is the top of your epiglottis, the top of your larynx. That's the word I'm looking for. Top of your larynx rise up as the epiglottis covers down, so it moves up as the epiglottis comes down. What that does is it blocks the windpipe from getting food or water in or beverage into your respiratory tract. All right, upper esophageal sphincter opens. Then the lower esophageal sphincter opens, and that's just above the stomach. Heartburn occurs when acid from the stomach moves upward past the lower esophageal sphincter, so LES, the lower esophageal sphincter, and damages the esophagus, usually due to overeating. So what's going on is there's no bicarbonate buffer, there's no mucus really in the esophagus, so it doesn't have any protection. So when some of the acid from the stomach comes up, it burns the esophagus, and it feels like your heart is burning because your esophagus goes right behind your heart. So I call it heartburn because it's happening right there where your heart is, but just behind it. Overeating, what's happening is your stomach is churning and squeezing, and too much pressure builds up when you've overeaten, and some of that chyme-containing acid squirts up into your esophagus, causing acid reflux or heartburn. Let's see, we've got someone in chat. Hiatal hernia, yes, it is dangerous. So one of the reasons why hernias are dangerous is because what happens is some of the small intestine pushes up through a gap. And this is especially true for men, because the testicles come through a small opening in your diaphragm. But the hiatal hernia, what happens is some of the small intestine pushes up through a gap there and then gets stuck. And the reason why that's a problem is because it causes anoxia. And that means the blood can't get to that part of the small intestine and it dies and can become infected. So yes, any kind of a hernia can be problematic, especially due to the fact that it can lead to
Strangulated hernia.
What's that?
Strangulated hernia.
Yeah, it's the hernia is what they do. What it basically means, you're not getting oxygen there. That tissue starts to die, and it can lead to potentially a blood poisoning or an infection of the blood. But either way, it's a problem that does need to be addressed. All right, the stomach, main function of the storage and sterilization of food prior to digestion. So it doesn't do a big job of digesting. Whether the secretions or excretions is going into the stomach, which is outside the body. Hydrochloric acid lowers the pH, increasing acidity, sterilizes the food, and softens it by unfolding the proteins. Lipase, 10% of fat digestion occurs here. So there is a little bit of actual digestion of fats in the stomach that happens. Pepsin is an enzyme that digests proteins, and it does get activated by hydrochloric acid. So when it first is released, it's what's called pepsinogen, which means it's inactive. The hydrochloric acid converts it from pepsinogen into pepsin, and now it's ready to digest. Now, you don't want the pepsin active in the cell that produced it, because then it would digest that cell in which it was being produced. So you produce it in the inactive form, it becomes activated inside the stomach, and that way the cell that produced it is protected against its own action. Okay, so what else protects the walls of the stomach? Bicarbonate buffer. It absorbs hydrogen ions or protons. You find it under the mucus, and it prevents the body from digesting itself. If you don't have an adequate bicarbonate buffer and mucus, you get what is called a, God, my brain just blanked. Oh my goodness, help me out, guys. When your stomach, get a hole in your stomach.
Gastric ulcer.
Ulcers, yes, ulcers are when your body starts, a hole in the stomach forms. Now, most people believe that ulcers are caused by too much acid production due to stress, but it turns out it's not true. But this shows you the mechanism by which hydrochloric acid is produced. So you produce the hydrogen ions in one part, release chlorine ions in another. So you're not producing hydrochloric acid in these cells, you're producing hydrogen ions and chlorine ions, and together they form hydrochloric acid so that the cell doesn't become overly acid, the cell that produces it. Okay. So this is what's going on here. Notice we have the gastric juice here, we have a mucus layer, and then we have the bicarbonate buffer underneath the mucus layer. What that does is it keeps the acid from interacting with the cells and destroying them. So it's the mucus layer plus the bicarbonate buffer protects you against autodigestion or damage from the acid. Okay, ulcers. The least common, it turns out, is too much acid production. So when people have ulcers, it's not the most common that is too much acid. The acid destroys the stomach lining. So if you do have way too much acid, it's called Zollinger-Ellison syndrome, and that can cause destruction of stomach lining, which is a problem because then those acids can start to damage other organs nearby. So ulcer, other organs, and that usually is treated either with medicines or surgery. Most common, believe it or not, is aspirin, which is acidic. It is acetyl salicylic acid. And that's why people who take a lot of aspirin often will take buffered aspirin. And bacteria, Helicobacter pylori, live in your stomach and can actually form a infection, which can eat through the walls of the stomach. And these are the two most common reasons people have ulcers, not too much acid. Now, it can also be due to stress. The stress causes a drop in your immune system, which can allow these Helicobacter pylori to grow if your immune system is depressed due to stress. What are some of the treatments? In the past, antacids. It gives you temporary relief by absorbing the hydrogen ions, but the problem is it can lead to what is called alkalosis. So producing a lot of acid and then you absorb it and turn it into some other material, it causes the body to become alkaline, which can lead to problems if you don't have a proper acid alkaline balance in your body. Today, drugs which block acid production, for example, Tagamet. What's another drug that blocks acid production? Tagamet and
Omeprazole.
yeah, I was thinking about the common names. Tagamet,
So like Nexium or
Pepsid, Nexium. You guys know the drugs that will block acid production, right? So instead of just neutralizing it, now you can block acid production if the issue is too much acid. Okay. And so I gave some examples, Tagamet, and you guys said some of those. All right. Intestinal phase, it did it again. All right. Secretin is a hormone that's released when acidic chyme enters the duodenum. So this is basically a response to food making it into the small intestine. So what does it do? It inhibits acid formation in the stomach and slows gastric emptying rate. So the idea here is it's a negative feedback mechanism. What it says is, hey, now we have some chyme in the duodenum. You need to stop putting more in so that I can digest this before we do any more. So basically, it just slows the process down when you already have some. It also stimulates the pancreas to produce bicarbonate buffer. This is important because you want to neutralize the acid coming from the stomach. So the chyme in the stomach is acidic, but the chyme in the small intestine is supposed to be slightly basic. Cholecystokinin, or CCK, is released when chyme containing fats enters the duodenum. What does it do? Another negative feedback thing. Inhibits acid formation, slows emptying rate. So slows down the release of food from the stomach into the small intestine. But more importantly, it stimulates the gallbladder to release bile, because what you're trying to do is break down the fats or emulsify the fats by the bile so that you can digest the fats that are in the food. Okay, pancreatic secretions. Into the duodenum or duodenum, digestive enzymes for all food types. Bicarbonate buffer neutralizes the acid from the stomach. And then into the blood, we already talked about this. This is the endocrine mechanism, insulin, which is what you secrete right when you're absorbing sugar after a meal, and then glucagon between meals to help maintain blood sugar. All right, why does blood go from the digestive tract to the liver before moving to the heart? Well, the idea is we have a thing called the hepatic portal vein. So blood goes to the digestive tract, basically most importantly, your small intestine, and then it goes directly from there to the liver. Now, the main reason why that happens is so that your liver can absorb sugars from the blood and store it before it gets to the body, maintaining blood sugar levels. Guys, can you give me just about 30 seconds? I need to do something really fast. I'll come right. All right, I'm back. So that's the reason why we have the hepatic portal vein. So the liver can both detoxify anything coming from the digestive tract as well as store carbohydrates before it reaches the blood. Okay, production of bile. Bile is produced by the liver and hepatocytes. Hepatocytes means liver cells. So interestingly, the liver does a lot of different jobs, but the cells all look fairly similar to each other. Bile moves through the common hepatic duct. You can see right here on the underside of the liver to the gallbladder. So it kind of makes a U-turn right at this V shape and goes into the gallbladder. Bile moves down through the common bile duct and fatty chyme enters the duodenum. So it goes from the gallbladder down into the duodenum or small intestine when CCK is released. So if somebody doesn't have a gallbladder, what happens is you can't store bile, so you can't have a large amount of it. So that means people who don't have a gallbladder might have some difficulty digesting fats.
Professor, what do you mean by CCK? What is that for?
Cholecystokinin, that was in a previous slide.
I just wanted to make sure I have clarification on what that abbreviation was for.
Yeah, CCK is cholecystokinin. That's released when fatty chyme enters the duodenum. That stimulates the gallbladder to squeeze, making the bile go down, then down into into the duodenum, and so it can break up the fat that's coming in from the food. Okay, fat digestion. Whoop. I guess this is just a picture. It shows you what's going on there. Oh, don't do that. Okay, so we have large fat droplets. We see the bile salts from the liver in the bile, emulsifying or breaking up the fats, and then the lipases break down these small bits into what are called micelles, which are tiny bits of lipid, and then the enzymes break that down into monoglycerides and fatty acids, and then it goes to the smooth endoplasmic reticulum, and eventually through the Golgi apparatus, and then to a structure called the chylomicrons that are released into the lymphatic system, and eventually make their way back to the large vessels above the heart. Okay, carbohydrate digestion, we see sodium co-transport of glucose or galactose. So we need sodium to move in as glucose or galactose comes in, but fructose comes through due to facilitated diffusion. Okay, amylase from the pancreas hydrolyzes complex carbohydrates into simple sugars. Brush border enzymes are enzymes that you find right on the surface of the intestinal mucosa, convert disaccharides into monosaccharides. Absorption, we have sodium glucose symport or co-transport that requires ATP. Fructose needs facilitated diffusion, so that is not active, that is passive. So you don't need energy to absorb fructose, but you do need energy to absorb glucose or galactose. And then to the extracellular fluid, and then to the capillary facilitated diffusion. Also helps move it from the extracellular fluid into the capillary.
So what is the mechanism of when someone is unable to break down fructose? Is it due to a lack of an enzyme, or is it due to the lack of being able to diffuse it?
Well, fructose doesn't need to be broken down because it's a simple sugar. So it might be the case that somebody could have a mild sensitivity to fructose, but you don't need to break down fructose because it is a simple sugar. And that's what you find in most fruits.
I'm referring to a particular issue called fructose malabsorption.
I'm not as familiar with that disorder, unfortunately.
Okay.
Maybe it's just that the facilitated diffusion mechanism doesn't occur as well as it does for most people. I'm guessing that's what it is, but it could be something else. I'm just not familiar with that particular disorder. I'm sorry.
That's okay. Thank you.
You're welcome. Okay, so let me talk about protein digestion and absorption. Now notice on the far right here, you can have polypeptides come through a process what's called endocytosis or transcytosis. So you bring it in, you move it through, and you dump it into the blood. This is how food allergies happen. The reason why food allergies happen is, and if this did not happen, food allergies would not happen. And let me explain why that is the case. It turns out that all foods contain the same amino acids. So what we're allergic to is not the amino acids in shellfish or peanuts or whatever. It's the fact these large proteins come in whole, they enter our blood, and our body overreacts to them. If we digested every single protein into individual amino acids, there would be no food allergies, because what would be entering your blood would be either dipeptides or monopeptides, which is amino acids, and that wouldn't cause an allergic reaction. But because it comes in this way, it happens. So the question is, why do we do it? There's two reasons why we do this. In infants, we do it so that we can absorb immunoglobulins or antibodies from our mother's colostrum and milk into our blood, giving us immunity. Secondly, we also bring it in so that we know what proteins might be coming in the form of invading microorganisms to help prepare the immune system to fight it. Unfortunately, some people overreact to that, giving them food allergies. And I know several people have food allergies against peanuts, some shellfish, things like that. It turns out some people have a highly sensitive IgE reaction mechanism that we'll be talking about in a later chapter. Okay, so this shows you how proteins are absorbed. I don't know why it's doing that, but anyway, okay, so water absorption, 9 liters of fluid into the small intestine per day. It reabsorbs about 7.5 liters a day of water. So it comes in very watery. It leaves the small intestine like very, very thick soup. About 1.5 liters of chyme move into large intestine. And then the large intestine absorbs about 1.4 liters a day. So that's almost all of that 1.5 liters. So you only lose about 0.1 liters a day in the feces normally. Now, diarrhea causes dehydration so that you can't reabsorb the water, so the feces comes out very watery. Now, why do bacteria want you to have diarrhea? Well, it turns out that bacteria spread in water, especially the ones like cholera that you get from drinking water. Now, imagine if a bacterium is embedded in a normal stool, which is solid, it can't escape. But if it causes the person to have diarrhea, when you go to the bathroom, it's all watery and the bacteria can exit your waste and go right into the water supply and infect other people. So that cholera causing diarrhea is on purpose. It helps it spread. Okay. So there you go. And osmotic diarrhea. Now, lactose can cause osmotic diarrhea, and olestra was a fat substitute that was common years ago that they stopped producing. Okay, this is an image directly from your book. It shows you the cecum, which is the beginning of the large intestine. No, don't do that. It shows the beginning of the large intestine. Then we have ascending colon. Why do they call it ascending? Because the waist is ascending or going upward. This is called the transverse colon because the food is going from right to left. Why do we say right to left? Because we're facing the person. It's not our right and left, it's the patient's right and left. So this is the right side of the patient, the left side of the patient. And then the descending, because the waist is now going downward. The sigmoid colon is where compaction occurs, the last water absorption. And then the rectum is where feces is stored until defecation. Okay, bacterial fermentation. Bacteria ferment vitamin K, and vitamin K is really important for clotting our blood. So having healthy bacteria in our large intestine is important. Digestion of certain substances by bacteria generates gases. So some people who are lactose intolerant can get gassy because lactose enters the large intestine undigested, and bacteria digest it, releasing methane, which is the gas that comes out of this. And beans, as we all know, can cause gas. It turns out that many people don't digest beans fully. So when those proteins enter the large intestine, bacteria get a hold of it, they generate methane, again, more gas. And the same with some other vegetables. Okay, immune defense. M cells engulf antigens from intestinal lumen. So basically they're absorbing these antigens or proteins from the intestinal wall and pass them to white blood cells inside the body. Macrophages and lymphocytes then receive the antigens and may initiate an immune response. So the idea is you're trying to pick up these whole proteins, which could indicate the presence of invading microorganisms. You want to alert the immune system. Irritable bowel disease or irritable bowel syndrome, IBS, is chronic inflammation of the gut due to normal GIT contents. Now, there is a review question that says irritable bowel disease or irritable bowel syndrome is due to unusual gut contents. That's false because it says normal gut contents can cause IBS or IBD. Okay, so remember for that review question, the answer is false because it's normal gut contents. All right, vomiting, scientific term for that is emesis or reverse peristalsis. It's contraction of the stomach muscles and reverse peristalsis of the esophagus. The idea is your body doesn't want anything in the stomach because there's either something bad in your stomach or you're stressed out, you're in shock, whatever. The body does not want stuff in the stomach at that point. It can be caused by chemicals in the blood, for example, alcohol. Some really severe pain can cause vomiting or vertigo. For example, seasickness, car sickness, spinning, things like that can cause it. And nobody knows exactly why vertigo causes vomiting. I think it's just because there's a disconnect between what the eyes see and the inner ear feels, and the result is vomiting or being nauseous, nausea. Now, if you vomit too much, it can also lead to alkalosis, because remember before, if you neutralize too much acid, it can cause alkalosis because you're losing hydrogen ions this way. But vomiting, you're physically just losing them from your body, and you can have insufficient hydrogen ion production. You can also get esophageal ulcers and tooth decay because you're not supposed to have acid in your esophagus or in your mouth, because the teeth are basically just rocks, and rocks dissolve when you put acid on. Okay, that is it for the lecture. But what we'll do now is we will review for the exam, which is coming up, and that is exam 5. That's chapters 22A through C is what I have. Do you guys have those review questions done?
A good chunk of them.
Okay, well, we're going to be going over them today, and any from last week that you may have, that it's basically 22A through C. So whatever chapters you have of those that are done, you've done review questions, you want to ask questions, let's go ahead and begin with 22A. So please get out 22A, look at your review questions, find any that you either couldn't find an answer to or want to double check your answer. Who would like to start us off 22A? Anybody 22A?
Could we do number 7 in the exchange of
Read
oxygen?
number 7. Wait, you got to say the number and let everybody find it quickly and then
Okay.
read it. Okay, so we're doing number 7 and now read it.
In the exchange of oxygen and carbon dioxide, the job of the lung surface is the opposite of that of the body tissues.
What do you think?
I think it's false, but I don't know.
No, it turns
I
out
put
it's
down
true.
true.
True,
That's true.
because in the lungs, you want oxygen in and CO2 out. And in the body tissues, we reverse that, right? We want oxygen out of the blood and CO2 into the blood. So that's true.
Okay.
So the
thank
lungs and body tissues are the opposite in terms of the movement of oxygen and CO2. Okay, let's do another one from 22A.
Can we do number 16, please?
16, read it, Catherine.
The main way that gases are different from liquids is that gases can be wet, while liquids cannot.
You get that one? Anyone get that one?
you. I got compressible and pressurized.
Compressible, right. So can be compressed. So the answer is they can be compressed, liquids cannot. Okay,
Thank you.
good. You're welcome. Anybody else from this chapter section?
What about 17?
17, read it, please.
The main way that gases and liquids are similar is that their flow is most easily restricted by.
Okay, how do we restrict the flow of gases and fluids?
Resistance?
Right, by increasing the
Okay.
resistance. And how do we increase resistance?
Decreasing the volume.
Right, decreasing the diameter of the tube. So both answers are correct. Increasing resistance and decreasing the diameter of the tube through which the fluid or gas flows. Does that make sense?
Yes.
Okay, good. All right, any others from this chapter?
Can we do number 8?
8, read it, please.
Extension question. If oxygen is absorbed by the blood in the lungs, how is mouth-to-mouth resuscitation an effective way to oxygenate a person with respiratory failure?
Okay, so the question is, how is it that mouth-to-mouth works? I guess most people don't do it in the traditional way anymore, but it can be done. So the question is, if you're absorbing oxygen from the air, when you exhale, how does that help the person? Well, 2 really important things. Remember that all the air in your airways is still fresh. You haven't exchanged any gases that, and all of that air is the first air that goes into the person. Secondly, even if you absorb a lot of the oxygen out of the air, having some is better than none, isn't it? So not only is the air that first enters their airway completely fresh because it came from your trachea and mouth, not from your alveoli. So that's completely fresh air. That's the first air to go into their lungs. And even the second bit of air that comes in, which is a little bit stale, still has oxygen in it. And that's why. By the way, that's not a test question, but I thought you guys might be curious about that. Okay. So there you go. Any others? 20,
I'll go with one more. 10.
10, read it.
One of the main jobs of respiratory system is to produce speech, true or false. And I was questioning that because it's not so much of a main job, but it is a function of it.
Well, if it's listed as one of the functions, it is a main job.
Okay.
So it's true. Yeah, it's true. Go with true on that. Okay, anybody else?
Can we do 34?
34, read it.
What molecules are released by mast cells during an allergic reaction? Was it histamines?
Histamines is the answer. Histamines.
Okay, I just wasn't sure.
No, it's okay. That's why we double check these, right? It's good.
Yeah.
Okay, who else would like to either double check or get one they couldn't get?
Can I double check number 9?
9, read it.
How does the respiratory system regulate blood pH? And I put by removing CO2 levels.
Right,
By
because remember,
removing
CO2 combines with water in our blood to form carbonic acid. So by removing CO2, you're also removing acid.
Okay, thank you.
Okay. Last one from this chapter, and then we got to move on. Who would like to do the last one from A?
15.
15, read it.
The alveoli have a significantly larger surface area than the rest of the respiratory system. True or false? And I put true.
True, right. It goes up exponentially. Remember, we went into the millions of square centimeters versus hundreds. So yes, that is true. Okay, let's call it done for this A, section A. Let's go to section B, please. Find your review questions. Valeria, did we just do 15?
Yeah, I just wasn't sure if that was the number you guys just said.
Yeah, it was 15 and it was true.
Okay,
So
thank
please listen for the question number when the person says it so that you can find it in your notes. Okay, let's go to B, section B. Who would like to start us off with section B?
Can we do number 8?
Read it, Catherine.
Residual volume can be directly measured using a spirometer.
What do you think?
I saw true based off of the information you had in the packet, or I was thinking true. But when I had done further research online, it had indicated that that was not one. It could
No,
be-
you cannot measure residual volume with a spirometer because that's the empty space in your lungs after you've breathed out completely. That has to be estimated based on gender, size, and other factors. So that's false. It cannot be directly measured.
Thank you.
And I believe that's in the notes. Anyway, let's
Well,
go
in
with
the notes,
another one.
it was making me think that it was true, and that it was true, and that's why I was questioning it, because like I said, I had done some other research that had indicated otherwise.
Yeah, it is false. It cannot be. Wait, what does the question say exactly again?
Residual volume can be directly measured using a spirometer.
False. It cannot. False. Okay. False. All right. Let's do another one from B.
Oh, can we do 9?
9, read it, please.
So since residual volume can be directly measured using a spirometer, 9 is basically saying that tidal volume can be directly measured using a spirometer.
We said residual volume cannot. Residual volume cannot.
But tidal volume, but this one is about tidal volume. Now.
Right. Remember, of all the volumes that we talked about, residual volume is the only one that cannot be directly measured using a spirometer.
Oh, so that's true then. Okay.
So all the rest of the volumes are true. You can use a spirometer to measure them. Okay.
Okay.
All right. Any others?
Can we do 15?
15, read it.
The liquid that is secreted by the alveoli in order to prevent the surfaces from sticking together is called, and I thought it was surfactant.
Surfactant is correct.
Yeah.
Thank
Surfactant.
you.
That was 15. All right. Anybody else for B?
19.
19, read it.
The amount of blood flowing around an alveolus is called
Perfusion.
perfusion.
Perfusion, yes.
Okay. Thank you.
You're welcome. Anybody else for this section?
Can we do number 16?
Well, wait, Alan asked, can you directly measure total lung capacity as well? No, you cannot, because there's one part of total lung capacity you cannot measure, which is residual volume. So total lung capacity also has to be estimated based on spirometer measurements and gender and body size, et cetera. So Alan, you cannot measure total lung capacity directly. I'm sorry, I cut you off. Who was saying something?
16.
16, read it.
The liquids of alveoli and pleural membranes have similar function, and I put false.
False, because remember the pleura, you want to be sticky to keep the lung stuck to the ribs. But inside the alveolus, you don't want it to be sticky because you don't want your lungs insides to stick together. So they're opposite functions. Pleural fluid and surfactant have opposite functions. Okay, good. Anybody else for B?
Number 14.
14, go.
The cells of the alveoli must be blank in order to facilitate diffusion of gases.
Okay, primarily I was going for moist, but they also have to be thin, right? So if you see two answers there, don't remember for Fick's law, we went through Fick's law.
Yeah.
It has to be thin as well as moist or wet. Now, here's the question. What if you only see one of them and not the other one? Should you reject the right answer because you're expecting two?
No.
No. The only time you reject a right answer is if there's another right answer and a third answer says two of these are correct. Please remember, everybody, that if that choice is offered, two of these are correct or all of these are correct, that's usually the right answer, and you should have a very, very good reason for rejecting it, not I wouldn't have thought of it that way, or it doesn't say so in the notes. I'm not testing you on what it says in the notes. I'm testing you on can you think critically, okay? So don't reject those multiple right answer ones unless you have a very good reason to do so. I'm just warning people again because they're still doing it. When I review old exams, still that issue is still coming up, okay? Let's do a couple more for this chapter, then we'll go on to C.
Can we do 28?
28, read it.
Typically, ventilation matches perfusion in the lungs.
True.
Okay. Okay, thank you.
Because we talked about ventilation. See you later, Matt. Have a good one, guy. We talked about ventilation perfusion mismatch, and that's a pathology that's not common. Okay.
Could I get help with 25?
25, read it.
What two structures work together to help breathing occur?
Lungs and diaphragm, no?
Rib cage and diaphragm?
Rib cage and diaphragm. Remember the lungs are completely passive. So it's the rib cage, intercostals of the rib cage, and the diaphragm work to allow the lungs to open, expand, and contract. Lungs don't have any muscle tissue at all in them, other than those controlling the size of the bronchioles. So the lung is completely passive in the respiration process, in the breathing process, okay?
Okay, thank you.
All right, good. Last one for this section, then we move on to C.
Can we do 26?
26, read it.
Oxygen and carbon dioxide move into or out of the blood independently on the concentration of the other gas. True or false?
True. They don't interact with each other at all. One's coming in, the other's going out. It doesn't matter how much of the other is there. Oxygen's going on its concentration gradient. CO2 is going on its own gradient as well. Okay. So I'm going to call it with section B. Remember, if you do want to send me some review questions through an email, do it as soon as possible because I may or may not be checking email later in the week. And please remember, you do have to have an answer to all the review questions you send me. And the answer has to be somewhat close. You can't just throw a Mickey Mouse answer in there and hope I'll give it to you. Any questions without answers will be returned unanswered. All right, so let's go to C. Who would like to start us off with section C of the notes?
Number 5.
5, read it, please.
Gases always diffuse in what direction?
Okay, what direction do gases always diffuse?
In or out? I don't know
They
if that
go
was
in
the right
some places,
word.
now others, but they always go what direction?
From the higher concentration?
That's right, Catherine.
Oh,
From
okay.
high concentration to low. They always go from high to low concentration. Good,
Okay, from high to
nicely
low.
done. Okay, what's our next question? Somebody bring it up.
Can we do number 2?
2, read it.
The movement of oxygen into blood is dependent on movement of carbon dioxide out of the blood. And I
What
put
did I just
false.
say earlier? They're completely independent of each other, right?
Yep.
So it's false.
False. Good, false. Okay, any others?
hold on. I thought I-
Section C.
Number 12.
12, read it.
What chemicals in the blood help prevent changes in blood pH?
What'd you guys get?
Buffers?
Buffers is correct. Buffers is correct. Okay, let's do another one.
Can we do 29?
29, read it nice and loud.
Blood is almost completely saturated with oxygen in alveolar arteries under normal conditions. And I got true.
True, right. It goes up to about 99%, 98 to 99%. That's almost completely saturated.
Okay, thank you.
Okay. Any others?
Oh, where did it go? I just saw the number. Oh, 27.
27, read it.
How many oxygen molecules can attach to a single hemoglobin cell? And I,
Hemoglobin molecule.
oh, molecule, sorry.
It's okay.
Was it? Oh, shoot.
4?
Did I put 4?
Four,
Yeah.
because there's 4 heme subunits, and that each heme iron atom holds the oxygen. So 4 is correct.
Okay, thank you.
Anybody else for C?
25?
25, read it.
Why does oxygen bind to hemoglobin in the lungs? And I
Because
put
it's-
because it's got higher O2 levels.
Right, high O2 levels cause binding, lower O2 levels cause release. Okay, good. Any others? We'll do a couple more, then I got to get going. One more? Sounds like someone's looking for one.
I think I'm good on that chapter.
All right. Hey, guys, I'm going to go ahead and get going. And since there was no test last week, we don't need to review any test stuff from last week. But next week, next Tuesday, we'll go over any questions that you had concerns of. Also, assuming I don't have an emergency at the end of my lecture, like I often do. But I do want to say best of luck. Oh, one thing I do want to remind you guys about, there's no more essays and there's no more subjective stuff for me to grade for the rest of the semester. So when you submit something, that is your current grade. So whenever you submit a lab, that's your current grade. Whenever you submit a test, that's your current grade. And that will go all the way through the final. So there's no more subjective grading on my part for the rest of the course. So you don't have to wonder what grade you have after you've taken a test. Will come up and that will be what you have. I'll be talking more about the final next week. But I do want to thank you all for coming today and being a part of this and participating.
Was Casey wanting question 10 on 22?
Do you, Casey?
I just see that there's a question on her side.
Oh, okay. Can you read it, Catherine?
In what situation would hypoxia occur but not hypercapnia?
Okay, so if you're holding your breath, you'd have hypoxia and hypercapnia because you're not breathing. But where's the situation where you're releasing CO2 but not taking in oxygen?
That's
Like
cyanide
you're-
poisoning?
High altitude, right? High
Yeah,
altitude.
high altitude.
Yeah, so you're getting rid of the CO2, but oxygen isn't coming in. Okay, guys, I got to get going. Good luck with this week's test. I hope you guys have a good rest of your week, a good weekend. Hope to see you all back on Tuesday for more lecture and a review of this week's test. Have a great one, guys. We'll see you next time.
Thank
Thank
you.
you,
Bye now.
Professor.
You're welcome. Bye.